The floor is yours, Alex. Please go ahead, take it away.
Yeah, thank you so much. Thank you guys so much for having me and giving me an opportunity to talk about this project, which I do love so much and care so deeply about. And although I am at Columbia right now, completing my master's and about to start my PhD there, this was work done at Indiana University with the help of Ameriflux. Um, and as I understand, this is the EnviroSensing clusters, and the focus is on a lot of sensors, which is also something very near and dear to my heart after a lot of experience working for small scale mesonets and biometeorology. However, on this project, we'll actually be focusing on this thing called the REESH project and how we've made site specific soil water retention curves to transform what we do measure with sensors into something new. We'll be comparing these REESH soil water retention curves to traditional pedotransfer functions. And then finally, we'll do a very quick introduction to two different applications of these curves and what we're hoping to grow the project into moving forward. So I won't delay any further and we'll kind of get right into it. So, as sensor people, I'm sure you are very familiar with volumetric water content, but if you are not, volumetric water content refers to just the volume of soil that is occupied by water. It's usually a simple percent measurement. And measurements of volumetric water content are incredibly popular. This is after early evolution of TDR technology, these sensors, which you can see on the lower left. And these sensors are not only very cheap, but they're easy to install and they have a long history within science. You can kind of see in this image here, that's me, my first job when I was 18, installing these sensors. And so many are quite familiar with them. And these data are additionally accessible because of data aggregation efforts such as Mesonets, Ameriflux, Neon, the National Soil Moisture Monitoring Network, and as well as remote sensing capabilities as well, such as SMAP or SMOS, which use microwave remote sensing to give broad scale estimations of volumetric water content. So the one thing you should bring away is that volumetric water content is very available, and because of that, it's often used as a proxy for drought stress in ecological models. These models describe the relationship between volumetric water content and ecosystem function as highly nonlinear, threshold dependent, site specific, can all be very complicated. And the reason it's complicated is because actually volumetric water content is not very informative of plant available water, which is an idea we're going to dive very deep into. And the reason for this is because soils are retentive. Soils hang on to their water. And different soils hang on to that water differently, and that's determined by soil structural and textural characteristics such as bulk density, organic matter, porosity, percent sand silt clay. All of it goes into this relationship that we refer to as the soil water retention curve. And that is that site-specific relationship between volumetric water content and matrix potential. Soil matrix potential refers to that tension at which water is bound within the soil. And it is incredibly influential on ecohydrology, in hydrology, everything. It is through difference in the pressure gradient from the soil through the stem to the leaves that allow trees to move water sometimes 50 feet up into the air. When soil matrix potential is too tight, when that tension is too tight, stomata will close, thus decreasing photosynthetic carbon uptake. When it is incredibly too tight, that's how we lead to cavitation and catastrophic embolism, thus leading to tree mortality. That same relationship exists within microbial communities as soil matrix potential dictates the flow of water and nutrients in and out of microbes. And it also, as I said, influences hydrological cycling, direct impacts on overland flow, infiltration. I could go on and on and on about how important soil matrix potential is. However, one thing it is not is incredibly available. There are in situ sensors, but they are not as frequently installed as the TDR volumetric volumetric water content sensors. And so often instead we rely on that soil water retention curve to transition our highly accessible volumetric water content into matrix potential. And we believe that this could have far-reaching implications throughout all of science. Some preliminary research done by Kim Novick, my old mentor, found that when we substitute soil matrix potential instead of soil water content in a relationship of, she said, your favorite flux or conductance. This could be evapotranspiration. This could be GPP, NEE. That threshold dependent nonlinear relationship that I mentioned earlier all of a sudden becomes more linear, more predictable, and easier to compare across ecosystems. And so the whole general idea is that by increasing soil matrix potential, we could increase our understanding of ecosystem flux and function, which is very exciting to us and kind of the basis of the Riesch project there. Now, this is not a new science by any means. Pedotransfer functions, which are empirical equations that historically have translated more accessible soil textural properties, such as percent sandstone clay, into the soil water retention curve. These have existed for decades. However, there are some issues when it comes to using these traditional pedotransfer functions. For one, many cannot truly capture site-specific influences of capillary and root matrices on that soil hydraulic. Many are constructed from, let's say, limited databases that do not include bulk density or organic matter, which are so important to that relationship. Or many pedotransfer functions are constructed from spatially inadequate databases. Let's just say, for example, they were trained off of only soils from Hungary, and then the result is a pedotransfer function that can only really perform well in Hungary. And this creates a big issue when we go back into those investigations on how soil matrix potential influence the ecosystem, because any uncertainty in your predictions introduced by the pedotransfer function then propagate into those predictions of ecohydrological processes. This is probably best explained by this figure here, also from Kim's paper, where she manipulated just one parameter in the soil water retention curve, and the variability introduced by that then propagated into predicted measurements of matrix potential, water content, and evapotranspiration. So our big focus is how can we not only improve the accessibility of soil matrix potential, but perhaps improve the quality. And that is where we get to the Riesch project. This was started as a partnership between Ameriflux and IU, and our goal was to provide site-specific soil water retention curves to Ameriflux sites so that they can then transfer their long-running history of volumetric water content data and also use any other meteorological and flux data often measured at these sites and look at their relationship to that site-specific high-quality soil matrix potential estimate. And this project was incredibly exciting for a number of reasons, which I will kind of dive into. The first one being is that we are utilizing a community sampling scheme. So I used to, and now Trevor does, Trevor and Max, I had a little kit of sampling equipment that you can see in the top right there. And that was sent out to all of these sites, all of these Ameriflux sites across the country that range a great diversity of ecosystems and soil types. And once they arrive, the technician at that location is responsible for the sampling. We give them a pretty detailed sampling book, which they follow. And they are instructed to sample 4 pits at 3 depths to create a total of 12 samples, 4 at the surface, 4 at 10 centimeters, and 4 at the level of their deepest volumetric water content sensor, which is often 50 centimeters, occasionally 30. We even had some sites return samples as deep as 150 centimeters, which my heart goes out to them. It is not easy to dig a 1.5 meter pit in the ground. So This really allowed us to have such a wide diversity of samples because we're no longer responsible um, financially and time wise to fly out to these sites, do all the sampling ourselves. Instead, we can really rely on this community effort. And in turn, each of these sites aren't responsible for buying the equipment, learning how to use it. It was all done at Indiana University. Here, I kind of illustrated and went a little bit in depth on how exactly these curves were made. First, we utilized the meter high prop system for the wet end of the curve. That one is pictured in the top right there. It uses the modified Schindler evaporation method. And what is nice about the high prop is these cores, I don't believe you can kind of see in the picture here, but the silver bands are the cores removed straight from the ground. So these are truly undisturbed, fairly large samples, especially large compared to historical methods of making the soil water retention curve. And on the base of the instrument, the black part there are two tensiometers which regularly measure water tension, and it sits on a scale which regularly measures that volumetric water content. And that continues until the two tensiometers cavitate. And at that point, we take two smaller subsamples from that big sample and we transfer it to the WP4C image just below, which uses a chilled mirror dew point approach. And this is more suited to the dry end. So it can accurately report water tension from -100 to 300,000 kilopascals. Um, the cool thing about the WP4C is technically it can measure the entire hysteric model, both the wetting and the drying front. But for the purpose of this research, we really only focused on the drying leg, and that was to ensure connectivity between the two. On top of that, we also measured saturated hydraulic conductivity with the machine called the KSAT, pictured on the left. And we also partnered with Rich Phillips' lab to get measurements of soil texture, root density, organic matter for every single sample as well. I've also included a picture of what the in situ soil matrix potential sensors use, which were distributed to some of the Ameriflux sites, but that analysis is not included in this presentation. On the left here, you can see these are what the data look like just straight off the machine. And there is obviously some visible noise there, especially as the sensors kind of reach the threshold of where each perform best. The connective part is the high prop. On the dry end, that's the WP4C. We then push those through a series of step tests to pop out the kind of clean curves, which are then fitted with the Van Canoochen equation. In this analysis, we'll also be comparing against two different pedotransfer function derived curves, each made using the same pedotransfer function, the Rosetta function, but with two different textural inputs. One being the texture that we found in the lab, and the second one being from a soil grids database. And this is to mimic what it would be like if I was just some eco-hydrological researcher out there who didn't have access to any soil sampling, any data. That would be the approach I would take. I would go online, find this database, select for my site, use the pedotransfer function. And so to go right into the curves, this is all of it together, but it's kind of a lot. So we'll break it up a little bit and take it step by step. First, just looking at the texture, on the left there being that database texture that I mentioned, and on the right, our texture that we found in the lab. And using the database really normalizes the texture across sites. You can especially see that in some of the sandy sites where we found sand percent to be upwards of 70%. They're now being classified by the database as a sandy loam, sometimes even a loam. And we also see very little change in texture with depth in the soil database as well. They report taking samples at different depths, but that information is a little bit harder to gain, especially versus our site where we know explicitly we have the surface, the 10, and the bottom measurement. When we compare how these textural inputs look when put into the Rosetta function, you can see the impact that that normalized texture has. On the left here are the petatransfer function derived curves with that soil database information. And all of these curves, even amongst the different sites, not only kind of show the same shape, but a very similar spatial distribution as well, which starts to break up a little bit as we use our lab-derived soil texture measurements. It kind of increases that spatial variability. But once again, they're kind of all a very similar shape, all glommed together there, especially when we open it up again and you can really compare to our Riesch lab-derived curves, which span the whole range. These sites are colored, and we'll kind of go to the next slide. The sites are colored based on the ecosystem type, just for easy comparison between the two. We have in blue and purples our forested sites. In the middle here are some dryland and desert sites. And then on the right are agriculture and rangeland sites. But this classification is quite arbitrary because within each selection, the curves are quite unique, quite diverse. And that all comes down to the unique interplay that we see going on between their individual textures, their individual organic matter contents, their bulk densities, and so on. So that really emphasizes the importance of that site specificity, and places a lot of importance on the work that we're doing there. To kind of continue that analysis, and these figures here, which are a little bit jumbled, I'll say, we'll kind of tease them apart a little bit later. You can see the difference on the right. On the top right are the lab-derived curves with the soil database petatransfer function. These are just the absolute differences, and you can kind of see the differences become smaller as the magnitude of the measurement comes smaller. So then we also, on the left here, looked at the percent difference. And then the bottom measurements would be the lab derived with the lab texture petatransfer function. And we can make some general conclusions here as well, but I would say the most important thing to notice is that the extent to which the site-specific curve varies from either petatransfer function curve is controlled by soil textural and structural properties, specifically soil organic matter. We don't have it distinguished on this figure here which sites are high in organic matter, but just trust me when I say, if a soil is high in organic matter, it's probably not going to be correctly represented by the petatransfer function, which makes sense because organic matter is not an input in the petatransfer function. It is just not taking that into account. Another very unique part about this project is because we are sampling at different depths, we can investigate how changes in soil textural and structural properties are also incredibly site-specific with depth. Even something as simple as organic matter, which is in the lower left here, we can, most of the time, we assume organic matter will decrease with depth. At this site, it increased with depth. So as I said, it's incredibly site-specific. And the impact that has on the resulting soil water retention curves mean that it is site-specific whether or not the retention curve varies with depth. Once again, both of those slides, how the curves compare to the petatransfer function and how they compare at each depth, is probably best represented in these figures as well. Just looking at the curves as is. This is just a slight selection of sites, but it kind of emphasizes everything I've said to this point. Each site demonstrates a unique pattern. Each site demonstrates a unique relationship. And so that kind of brings us to the key points of that first section there. Although singular characteristics can predict general trends in the soil water retention curve, i.e. sandy sites might look like sandy sites. Ultimately, it is the unique interactions between site-specific factors that provide the entirety of that curve, emphasizing the importance of this project. And the second one being the Riesch protocol offers a unique perspective into site-specific factors influencing soil water retention, influencing how the curve might look with depth, so on, so forth. And these cannot be captured by the traditional pedotransfer functions. Moving on to the second part of this presentation, I'm going to kind of go into some of the applications. I'm going to bring you back to that idea that I mentioned earlier in the talk that research often describes the relationship between soil moisture as site-specific, nonlinear, threshold-driven, and the idea that when we replace soil moisture content with soil water potential, we now see stronger links to ecohydrology. And so that led us to the question of like, can prediction of ecosystem scale gas exchange, i.e. eddy covariance, be improved when using either in situ volumetric water content, the lab-derived soil water retention curves, or our two pedotransfer function curves? This idea was supported by a quick preliminary analysis I performed a year and a half ago at this point. Just terrifying thought. And in this, we were looking at tower-derived GPP estimates and tower-derived volumetric water content measurements, in situ volumetric water content measurements, and then matrix potential measurements, which were transformed using the Riesch curves. And you can see, as I stated before, that when we use matrix potential instead of volumetric water content, the relationship is more linear, more highly correlated, and it is much easier to compare across these ecosystem types as well. This analysis was continued by my mentor, Daniel Beverly, as we prepare our paper, where instead of using tower-derived GPP estimates, he was looking at normalized surface conductance. And as we added more sites, that signal and that message does get a little bit more noisy, but the main message is clear. Out of the 10 sites for which water potential, out of the 10 sites that we looked at in this analysis, water potential outperformed volumetric water content in 80% of them. And so that relationship between surface conductance was stronger using matrix potential for 80% of sites, specifically using the lab-derived water potential. However, in some of these sites, specifically the sandy sites, that was not true. We are currently investigating why that may or may not be so. It might be reflecting the Van-Genoochen parameters and the ability of the Van-Genoochen model to fit the sandy sites. It might be reflecting the ability of the equipment to handle sandy sites. We are unsure, but we're excited to continue looking into that as we move forward. The second application that I am currently exploring as part of my master's thesis kind of evolves from this main idea that while we are so proud, and I spent this whole presentation talking about how great these site-specific water retention curves are, they're still fairly hard to make. It takes a lot of time, it takes a lot of labor. We were fortunate enough to have an entire lab and multiple technicians devoted to this, but it's not necessarily feasible to expect that of every single site who is looking to use matrix potential. This is exacerbated by the fact that at this stage, our sampling is restricted to Ameriflux sites. And so I had very many people coming up to me at the multiple presentations I've given on this project asking, how can I do something similar? And I didn't want the answer to be, sorry, you can't, and or sorry, go ahead and buy a high proppant WP4C for this many tens of thousands of dollars. And so we are now turning back to the pedotransfer function using a neural network. Neural networks have a long history of being used to develop pedotransfer functions. The Rosetta pedotransfer function that I showed earlier is actually an example of a neural network pedotransfer function. And that is because the neural network does such a good job without prior concept of the relationships between inputs and outputs, and because it is so well suited for capturing complex nonlinear relationships. The issue and the reason why we can promote our site-specific water retention curves over those those traditional pedotransfer function was, I believe, because many of these pedotransfer functions were made using poor soil water retention curve databases. And many of them were made without using the full range of soil structural properties that we know is important to this relationship. For example, a majority of pedotransfer functions rely just on soil texture and ignore organic matter bulk density. And so now that we have the Riesch project and we have accessibility to high quality, high resolution curves like never seen before, we are looking to improve the neural network pedotransfer function and eventually planning on plugging this new pedotransfer function into large climate models to see if it can improve their performance as well. We're very, very early in this work. As I said, I'm completing this for my master's thesis with Dr. Linnea Hawkins from NCAR. However, is just another idea of one of the many avenues we're now able to head down with the Riesch curves. And so that kind of leads me to the wrapping up stage of this presentation. As we move forward, we have a couple next steps. Those still back at IU, Trevor and Max, are continuing the Riesch project. We're hoping to finish 40 to 50 Ameriflux sites. And I believe we are getting to the halfway point on that. I could be wrong. And there's even a possibility to include and expand this analysis even further past this point. This data will also be publicly available on the Ameriflux sites via each site's unique DOI. And so when you look at the applications for which we are using this, we are hoping that many other people will come up with their own applications as well. And we are also finishing up the first paper that kind of summarizes everything that I went over today as well. That paper is close to submission, so keep your eyes out loosely, like maybe in a little bit more time, like really keep your eyes out, but we're super, super excited for that. And I will just kind of end off on this with a lot of acknowledgments. As I said, this is a community sampling event. So I really could not have done this with all of the contributors, the site PIs, especially their technicians, those people digging those holes. Thank you so much. Thank you, Max. Thank you, Trevor, and the technicians at Indiana University that have helped accumulate this data. Thank you to our funders at Ameriflux DOE. And thank you so much to Dr. Daniel Beverly and Dr. Kim Novick for helping me. I definitely would not be anywhere near to where I am today without them, and this project. I've also included at the bottom of this here my email if people have any reason to reach out. And then we will also have, I believe, perhaps if anybody has questions, time for those as well. But other than that, I'm finished. Thank you.
That was great. Thank you, Alex. Fantastic. Really fascinating stuff. And there was so much from my work with sediment transport monitoring, surrogates for that just rang out in your work here. So I found it very exciting and yeah, just really great stuff. I will open it up to the floor first for questions, and then I'll ask some that I have at the end. Does anybody have any questions for Alex?
I don't have a question, but I do have a comment. Great work, Alex, of course. Not surprised at all. In terms of the performance in sandy soils and it not being improved with the lab-based soil matrix potential and water retention curves compared to volumetric water content. I never worked in deserts until I moved to Las Cruces, and so I was out there taking those samples that I sent back. And I wonder if it just has to do with, I mean, there's just no structure to these soils at all.
No.
And so even at depth, like we were trying our best to keep it at that depth, but it just all falls apart. I mean, I felt like I was in a sandbox. And I wonder if that has anything to do with that lack of improving that performance, because at least with the TDRs, at least they're just in there, you know?
Yeah, I definitely think it's a big problem. In general, I feel like there is a lot of improvements to be made in the way that we understand soil moisture in these sandy sites, even with the TDRs, even with any in situ sensors as well. Um, a lot of Ameriflux sites receive those in situ matrix potential measurements. And then when I ask sandy sites for that data, many will not even share them with me. I think because they're worried that the data is bad, but that's not reflecting anything they've done. That just reflects that we don't have the means at the moment to understand the moisture dynamics there. It's just so flashy. It's so difficult. So thank you for the comment slash question.
And I promise we didn't seed that, right? Because that's a great lead into this group. There's a lot of innovators in this cluster that'll see the recording too. Folks, there you go, right? Like there's a spot to focus on, right? That dry section. Interestingly enough, yeah, so it's really interesting to see the curves respond like that, and then like the structure itself just fall away. Um, thank you, Sandra. Anybody else? Any other comments, questions? Okay,
Oh, I
I'll
see a
ask.
question in the chat.
Oh, yes. Scott had to sign off. Excellent. 24 sites. Man, that's hard work. Digging pits like that, I know. Whew. Yes, so here we go. If you have variation in the results from different sites with sandy soils, it may have something to do with whether soil fungi like
Likely
microhazel.
mycorrhizal.
Yes, yes,
Yeah,
excellent. Thank you. Um,
yeah,
Martha,
I
no,
definitely think
save me here.
so.
Yeah, that well, I did a postdoc at the Face site in Nevada, and when it was really, really dry in the late summer down there, and this is outside of Las Vegas, so very much desert. Um, you would get the sandy soil would just crumble apart. You couldn't get anything. And then when well, we were looking at mycorrhizal fungi somewhat, but when it would be more present after rain, and part of it's probably water, but after rain, you'd get these gluey glycoproteins and the sand would stick together more. So, kind of interesting there. There's probably some cool interplay, moisture, glycoproteins, fungus, all that stuff. So, yeah,
Yeah, that is super cool. It is, so it is at the wet end of the curve. That is what is dictated by soil structure. And so it wouldn't
yeah,
even just be the aggregation
right.
caused by the glycoproteins, but you would also
Yeah,
have impacts from porosity via all
sure,
of that
sure.
and whatnot. It's
All
a
that
truly
stuff.
complicated system.
Yeah, with feedback loops.
Yeah,
Nice.
that's super cool.
Interesting stuff. Thanks for your presentation.
Thanks, Martha. I had never thought about like biofilm increasing decreasing porosity to slow flow, right? While it rains in a desert. Man, biology is cool. It's really neat. Thank you, Martha. I appreciate. Man, you're always like,
Oh, thanks.
get away from just dirt, Joe, and think about the bio. Fantastic. Thank you. So one of the things, this is an interesting topic in the USGS with these sensors, the TDR sensors and such. We we don't have a method for calibrating them at this moment. And I don't know if you know the researcher Todd Caldwell. He was connected with University of Indiana with some GS folks, but Todd was working with a neutron sensor, cosmic sensor for soil moisture. Um, I wondered if that had come across, if you had any kind of interaction with that, and then if you had any thoughts about calibrating an instrument in the field, such as that TRD.
Yeah. I do not have any any experience with the neutron probe soil moisture sensors myself, beyond just kind of the surface level understanding. And I also do not have any personal experience with calibrating. However, through my experience working at the soil moisture, at the Indiana Water Balance Network, which is a mesonet of sorts, we did commonly see the importance of calibration. Our
Mm,
sites, half our sites were calibrated, half weren't. And it was not always necessary. I believe it's recommended with certain soil types. However, I am not as familiar to make commentary on that. But I did do a very quick analysis at that time where I was working on flagging, QA/QC flagging of soil moisture data, and sensors that were calibrated were flagged much less often than those
wow.
that weren't. So it is incredibly important and always a question that comes to mind as well. Uh, I don't necessarily know what to do about it. We can't force people to calibrate their sensors, and it does become an issue
Yeah.
because at this point, I'm no longer a sensor installer, but I use other people's data, and I can only hope that they calibrate their data, but I do not know. That metadata is often unavailable.
Right.
So as you can see, I have many thoughts, but little to add.
Well, what the part that you added is significant. So that's fantastic. Maybe in the future we can reconnect on that topic. Just to dig into more of those experiences with the QA/QC, because that's what's holding us up now. Is we have very high stipulations on data uh, at times with certain cooperators, and how to verify it is the question at hand. So thank you for sharing that. We have about 2 minutes left. Anybody else have any questions? Okay, well, really appreciate you coming, Alex, and the folks from the field lab. Man, love it. Love seeing those pictures of the field work. I know for me, I always like getting those pictures in there, right? Because that in itself is reward for the field crews. It's like, yeah, we're out there digging 1.5 meter pits. That's pretty substantial. I
Yeah.
did. I enjoyed your use of the ternary diagrams and the coloring. That was very impactful, and it sorted it out nicely. So thank you for joining us, and please welcome. Come back to ESUP, and going to transition quickly into our summer meeting. It might be of interest. Um, we understand that we have to have the statement of interest in by the 15th. Uh, so James and Cassandra, let's try and organize quickly on that. And for the rest of the cluster, we're organizing around this soil moisture topic. So that's hopefully we'll continue that interest into what's that, July, right, Cassandra, for the summer meeting?
So July 20th to the 25th, maybe?
Excellent, excellent. So I yeah, man, I'm getting my butt kicked at work with all the silliness that's going on. So we're going to try and clear that out and make a little bit more time for ESA quickly, so that we can get in that session, because I think that's going to be impactful. And there's a ton of interest from the community. So if there's nothing else, I'll pause here. Yes, excellent work indeed. Thank you, ESIP May 2025 in the books. Appreciate everybody for coming in. Please join us in June. We have a fun session in June, right? This is going to be the lightning talks, the first one in a while that I think we've done. So we're looking forward to that. Couple of short presentations on different things. I'm going to show dirt fall. Super exciting. So tune in for that. But yes, no, thank you everybody. Great to have you, and we'll look for you next month.